Hello again. Okay, so in the third video, I explained what happens during oxidative phosphorylation. I want to emphasise the importance of oxygen acting as the terminal electron acceptor. The reason this is so important is because it allows the accumulated reduced NAD and accumulated reduced FAD to be reoxidised, meaning that the oxidised NAD and the oxidised FAD can go back to the previous stages of aerobic respiration, so back to the Krebs cycle, or back to the link reaction, or back to glycolysis, to be reused. Oxidised NAD, oxidised FAD are both needed to accept hydrogen and electrons from the substrates earlier on in aerobic respiration. Without oxidised NAD, without oxidised FAD, the process just stops. Okay? There is only a limited amount of oxidised NAD and FAD. So if there's no oxygen to accept the electrons from it, respiration cannot continue. So in this video, I'd like to explain how anaerobic respiration allows for the reoxidation, so the oxidation of the reduced NAD and FAD. Yeah, so I'm going to have to do rubbing off here. Okay, the first thing to say then, anaerobic respiration involves glycolysis only. And that glycolysis, you remember, occurs in the cytoplasm. So let's just remind ourselves of glycolysis. We start with that 6-carbon glucose, 1, 2, 4, 5, 6. It undergoes phosphorylation. So 2 ADP donate their inorganic phosphate, 2 ATP to form 2 ADP. And we get that 6-carbon, or the phosphorylated glucose, as it's sometimes referred to. There's a load of different names for this. We'll just refer to it as phosphorylated glucose for now. Okay, that then undergoes, or it splits into 2 trisophosphates. Each with their inorganic phosphate. They are then oxidised to pyruvate, another 3-carbon molecule. Remember, these are two trisphosphate, and then we'll have 2 pyruvate. Now, this is where there are some problems. Okay. For this stage to occur, for trisphosphate to be oxidised to pyruvate, there must be a supply of oxidised NAD. Oxidised NAD is reduced by accepting hydrogen and electrons from the triose phosphate to produce that reduced NAD. And that occurs with each one. Okay. At the same time, this is an oxidation, so that provides a little bit of energy for 2 ADPs to be converted into 2 ATPs. By, remember, substrate level phosphorylation. This can only continue, or in fact, in the presence of oxygen, pyruvate will then pass into the matrix of the mitochondria, where it undergoes the link reaction, Krebs cycle, etc. However, if there is no oxygen to act as the final electron acceptor, we quickly, or the cell quickly runs out of oxidised NAD. This process then stops, and so there can be no more pyruvate, no more substrate level phosphorylation, no more oxidative phosphorylation. The cell is unable, the mitochondrion and the cell are unable to produce any more ATP. So there needs to be a way for that reduced NAD to be recycled, to reform, to be reoxidized back into oxidized NAD. Now this happens in one of two ways. Okay. In animals, the pyruvate will accept the protons from the reduced NAD. Okay, so let's show on this side what is happening in animals. Pyruvate is reduced to lactate, and that happens when the reduced NAD donates its protons and its electrons onto the pyruvate to form lactate. That creates more oxidized NAD. That oxidized NAD can then return to allow the oxidation of triose phosphate into pyruvate, and to allow more, that should be 2 ATP there, to allow more ATP to be produced by substrate level phosphorylation only. So this is in animals. Now I'm not going to draw this all out again for some plants and fungi. I'm just going to have to, you're going to have to imagine that on this side of the diagram we've got plants and fungi. Okay, so some plants and fungi, instead of producing lactate, will produce first ethanol. So ethanol is produced when carbon dioxide is released from the pyruvate. Okay, so ethanol is a 2-carbon molecule. Ethanol is then oxidized into ethanol. So this oxidation ethanol is reduced into ethanol by accepting. So, the ethanol is reduced to ethanol by accepting protons and electrons from reduced NAD to form more oxidised NAD, and that oxidised NAD is then available to accept more protons and electrons during the oxidation of triose phosphate into pyruvate. Okay, in both cases then, the importance here is oxidised NAD must be regenerated, must be recycled to allow substrate level phosphorylation to continue. This is anaerobic respiration. There are some issues with that. In animals, the accumulation of lactate or lactic acid lowers the pH. So this often happens in muscles that are respiring anaerobically, and that can cause muscle fatigue. As the pH is decreased, it has an effect on the sliding filament process for muscle contraction, causing muscle fatigue. So this can't continue for too long. In some plants and fungi, well, eventually the accumulation of ethanol becomes toxic, and therefore, as the concentration of the ethanol increases, it will eventually poison the plants and the fungi. I guess it does have some benefits. This is fermentation, so this is how things like beer and wine and cider are produced. Okay, so that is anaerobic respiration. Questions about this require you to make a comment to say that oxygen is not available as the terminal electron acceptor. Instead, pyruvate accepts electrons and protons from the reduced NAD. This regenerates, regenerates is fine. So regenerating oxidized NAD, allowing glycolysis to continue. And this is a really common question because it applies your understanding of aerobic respiration, because we know that aerobic respiration requires oxygen as the terminal electron acceptor. It applies your understanding of glycolysis, because we know that glycolysis can only continue if oxidized NAD is available. And it adds in this stuff about how that oxidized NAD can be regenerated. Check the question. Does it say a particular type of animal tissue? So it might say something about liver tissue, or it might say the root of a particular plant, or it might say something about biofuels, for example, with sugarcane. So depending on whether it's animal tissue or plant or fungi fungi could be yeast, for example. You need to think about exactly how that reduced NAD is oxidised back into oxidised NAD to allow glycolysis to continue. Okay, but a very common area for questions. I hope that is useful to you.